Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Kyle Hall from Business Review, and I will be your host today. It is our pleasure to have GVK Biosciences with us, who will be presenting this webinar entitled Challenges and Solutions in Handling In Vitro Assays. Today's guest speaker is Dr. Prem Kumar Arumugam, who is Associate Vice President in Vitro Biology. I'd like to welcome you to our webinar platform On24. You'll notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you received via email to rejoin the session. In order to ask questions, you can send them in via the questions widget. Type them into the box at the top left-hand corner of your screen and click Submit. We'll allocate some time at the end of the session to address any questions or thoughts that you may have. Please use the yellow help widget if you require any assistance, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But for now, please allow me to welcome Dr. Prem Kumar.
Thank you, Kyle, for the nice introduction. It's good morning, good afternoon, everyone, and thank you for joining this call. Today, I'm going to present novel label-free screening assays using rapid-fire mass spectrophotometry. And this is basically always for our organization, safety and compliance first before the business. Before moving to the assay part, so basically I just would like to describe about why this rapid-fire technology and how it is basically superior to the existing techniques. As you all aware that majority of these assays uses artificial labels such as fluorescents or radio labels, tool compounds, and this is what we will be using for screening assays. The issues with these type of compounds are basically it can alter the protein conformations or the target conformations, or these compounds can have an autofluorescence effect, or it can have an quenching effect. So this will lead to false positive or false negative data. To avoid this, the alternate would be a label-free approach. Obviously, label-free method would be that we will be investigating the target proteins in the native form. Label-free methods are basically currently people they use in secondary screenings. The major reason being that the throughput, because the currently available technologies like Biochemical or conventional LCMS methods, it's time-consuming and also labor-intensive and very expensive. So based on all these three reasons, people they use this as a secondary screening rather than a primary screening. So the solution for this is basically a rapid-fire high-throughput mass spectrophotometry system. This is a label-free platform that delivers both biochemical and functional assays in any type of biology matrices. Now I will be discussing about two type of assays that we built using this rapid-fire platform. One was basically measure and quantitate lipid, more precisely the prostaglandin PGT2. The another one is basically measures sterols in cholesterol pathway. So this, the first part actually I'll be discussing about the, how we built assays to measure prostaglandin D2, in short PGD2. Before we go into the assay detail, I just would like to go over the Prostanoid biosynthesis pathway. The left side is basically the pictorial representation of the pathway. The arachidonic acid, this is a substrate for cyclooxygenases. Basically, these arachidonic acid are present in the membrane, and this will be activated by phospholipase A2, and it releases arachidonic acid from the lipid membrane. Then in turn, arachidonic acid will be catalyzed by cyclooxygenases, and that it produces PGH2. The PGH2 is basically in turn converted to several prostaglandins. One such prostaglandin is PGD2. That's basically all the way the bottom one. And this catalysis is done by an enzyme called PGD synthase. Now, why PGD2? Now, PGD2 plays a major role in allergy and inflammatory processes. Inhibition of PGD synthase, the enzyme which is responsible for converting PGH2 to PGD2, will be a potential target for various immunological and allergy-related diseases. For this assay, we have used RBL cells, that is basically rat basophilic leukemia cells. Generally, it is basically mast cells would be the more appropriate cell system to use. But however, being mast cells as a primary cells, people adopt RBL cells for this purpose because it behaves like mast cells. It has very high expression of Fc epsilon receptor, and that is the receptor is basically that IgE anti-immunoglobulin. That basically these levels of immunoglobulins will get elevated during allergic responses that recognize this receptor, and through that it all pro-inflammatory responses will get elicited. So now, why we wanted to build a rapid-fire-based LC-MS assay for this? So current assay platforms like ELISA or fluorescence polysystem assays, predominantly they use labeled probes or antibodies. Now the issue with these assays are there are a lot of these antibodies-based assays has cross reactivity with the other prostaglandins like PGI2 or PGE2, TXA2. These are all very similar in structure. So there is a possibility that it can cross react. So we may not be able to accurately quantitate the PGD2. There is a recent methodology, it's been reported in the literature using LC-MS, although it is basically, it can accurately predict PGD2. But as I said, conventional LC-MS assay is time consuming and require more samples. And also it is basically high cost. To alleviate these issues, so we wanted to build a high throughput LC-MS assay using rapid fire. Now this is basically an assay overview. To build these assays, what we have done was basically we have optimized the rapid fire mass spectrophotometric conditions. Then we have optimized the cell density, and we also optimized the right concentration of calcium ionophore A23187, and this is basically a inducer for this pathway. Calcium ionophore elicit calcium, and the calcium in turn basically activate the phosphatase, which is basically responsible for activate the arachidonic acid. So that we have optimized the right concentration. Then actually we have also assessed what is the DMSO, the tolerance level for these cells and this assay. Then finally we validated this assay with a reference compound HQL179. And the test system used in this assay was RBL cells. So this is the optimized rapid fire condition. So for this, we used a triple quad 5500 mass spectrophotometry from SciEx, and this is basically coupled with atmospheric pressure chemical ionization APCI as a source for ionization. Now, I don't want to go into the details of those aspirations or the other elution conditions. This is all mentioned in the slide deck. The important thing is basically we used C8 cartridge for this assay. Now, coming to the mass spec conditions, the MRM. So we used two types of standards. One was basically the PGD2. It's commercially available standard. The parent mass is basically, it's mentioned here as a Q1 mass. It's about 351. This is what actually we detected in the mass LCMS. The major daughter ion for this PGD2 was basically 270.9. That is mentioned here as a Q3 mass. And for internal control, we used a deuterated PGD2. That's a D4 PGD2. And for this, the parent mass, the molecular weight was 355.1, and the daughter mass is about 275.2. So these masses are very specific that can detect only the specific analyte. So 270.9 is can detect PGD2. 275.2 can detect only deuterated PGD2. So using that conditions, what we have done was basically we wanted to assess the sensitivity of these assays using PGD2 as a standard. So what we have done here was basically we prepared a standard curve from starting with 19 nanomolar to all the way to the 10 micromolar. So if you look at the right-hand side, it is basically this is the standard curve coming from this rapid fire MS analysis. So we see clear linearity from all these the concentrations what we have tested. The left-hand side is basically the area under curve for this standard. So what we found here was basically we were able to see the minimal detection limit was about 70 nanomolar. That is basically the limit of detection for this assay in this cell type. So now before I move to the other optimization conditions, I just want to go over the general protocol, although it is basically here I have given a detailed protocol what we have optimized just for for a preview. So what we have done was basically in the day one we have seeded the cells, and the day two it was basically the cells were washed, and then we have ordered the test compound. Then it's basically the post 24-hour incubation. We have stimulated the cells with calcium ionophore. Then it's basically it was incubated for 30 minutes. Then we have pelleted the cells and taken out the supernatant, and this supernatant was basically assessed for rapid fire analysis. So this slide is basically about what is the optimal cell number and what's optimal ionophore concentrations. This assay gives this is basically this slide talks about. If you look at that this slide, what we have done here was basically we used different cell number from 12,500 cells to all the way to 100,000 cells, and we used from 0.25 micromolar A23187 ionophore to all the way to 4 micromolar ionophore concentrations. So what we found here was 50,000 cells gave the best, the maximal response, and 1 micromolar A23189, that's basically the gray color, arrowed in green, gave the best response. So what we have optimized here is basically the cell density. That's 50,000 cells, and the optimal concentration is 1 micromolar ionophore. So moving to the next slide. So this is basically using that the rapid fire optimized conditions as well as the cell number, the ionophore concentrations. What we have done was basically we assessed the DMSO tolerance. So we tested from 0% DMSO to all the way to the 4% DMSO. The highlighted one, the boxed one, the red color, is basically 0.5%. This assay was tolerating up to 0.5% DMSO. So using these conditions with the 0.5% DMSO, using 50,000 cells, we tested a reference tool compound HQL79 in this assay, and then we found an IC50 about 4.3 micromolar. Previously, this assay was done from the literature. It is basically they used a fluorescence polarization assay, and they reported IC50 was 6 micromolar. So it is well aligned with the FP assay data. So then what we have done was basically we have selected a few of the compounds based on the FP biochemical data. It was basically this assay we developed for one of the client, and they were actually doing in-house the biochemical fluorescence polarization assay. Based on that data, there are few of the actives and inactives compounds we have selected based on the rank order potency. Then we profiled this compound in this assay. What we found was basically the IC50 was very well. It was alignment with their in-house data. So that is basically 182 for compound X, and all the way this compound Z. It's an inactive compound, showed inactive, and active compounds, more active, less active compound, less active, like an FP assay. So with that, it is basically, so this assay was basically repeated at least 3 times, and this assay reproducibility was so great. So now what we have done was basically we wanted to adopt this assay for a biochemical as well. So then it is basically we have done this assay. This slide talks about the biochemical assay data. If you look at that, these IC50 information, it's basically the HQL79 compound. As we saw earlier, it's about 1.9 to 2 micromolar IC50. It was FP assay was about I think there's something 6 micromolar or something. Here it is basically there is some error, looks to me that. The other compounds is basically showed very similar trend like rapid fire assay. The compound X in FP assay was about 10 micromolar from client data, and for using the rapid fire it was about 28 micromolar. So if you look at that, this is also inactive become inactive and active become active. So these biochemical assays were very well. It's basically in alignment with the other assay format. The right hand side is basically the cellular assay comparison. Already I discussed on that. If you look at the rapid fire biochemical assay versus the cell-based assay, again the potencies were very well in alignment with the biochemical data. It's basically actives become more active, and inactives remain inactive with the cell-based assay as well. So with that, it is basically, so we have a very robust assay, and in fact, actually that assay we used to support one of the client program for almost 2 years. So now it is basically, I am moving to the second part of our discussion. It's basically the assay to quantitate, to measure various sterols. Now this assay primarily actually we built to support one of the anti-fungal program. So the left hand side talks about the general, the cholesterol pathway from all the way to the acetyl CoA, the cholesterol formation, it happens almost close to about 19 to 20 steps are involved in this one. Now the highlighted, the sterols, that is the third one from squalene. The lanosterol formation happens from squalene epoxide lanosterol cyclase. So that the lanosterol, and the another highlighted item is basically the dihydrolanosterol and the cholesterol. And these three sterols are actually, we built a method using rapid fire. So this program is primarily actually targeted against CYP51A1. If you look at in the pathway, CYP51A1 is a key player in biosynthesis of steroids and the lipids in eukaryotes. In fungus, it is basically ergosterol. Its ergosterol is important, the functional moiety in the cell wall. So it is basically, if you inhibit the CYP51A1, that affects the ergosterol synthesis in the fungus. That in turn, it kills the fungus. So even it is basically, this is being utilized in the agro perspective as well. So to support that C51A1 target, it is what we have done was we built methods for lanosterol, dihydrolanosterol, and the cholesterol. The biggest challenge of this measuring this sterol is, again, majority of these pathway these sterols are very similar in structure, and normally the conventional, there is no assays are available. Whether it is basically ELISA or any other assay formats are not available. The only assay format available to measure this is GCMS. Now GCMS is basically a very laborious technique and again time consuming and very expensive. To avoid that, what we have done was basically explored the possibility of building these assays in a mass spectrophotometry using rapid fire. Now this is basically an assay overview. Again, so these are all the various conditions that we optimize the rapid fire, mass spectrophotometry conditions, cell number, incubation time, DMSO tolerance. This assay is basically the current format for GCMS assays. People, they use 6-well plate. So actually we wanted to test a different plate format to improve the throughput and also reduce the cost. So that I will be discussing in the subsequent slides. The tool compound we used to validate this assay was oxyconazole. Now the test system we used was Eckert cells, and this cell is basically having endogenous cholesterol synthesis pathway. Now this is optimized MS conditions. Again, the same instrument was used, triple quad 550 from SciAx, and with APCI as an ion assistant source, and the cartridge used was a C8 column. The MRM developed for this is basically the cholesterol, the parent ion molecular weight is basically about 386.5. The major daughter ion, that is basically Q3, we found was basically 368.3. That's very specific for cholesterol. Now coming to the lanosterol, the parent ion mass was 426.5, and the major daughter or predominant daughter ion was 95.2, very specific to lanosterol. Now for dihydrolanosterol, it's 411.3 parent ion and 95.1 a major daughter ion. So this is the conditions that we optimized to accurately quantify the specific sterols. Now, so before going to those conditions, this is the generic protocol that is we followed. Eckert cells were seeded for almost for 24 hours. Then it is basically treated with a test or reference compounds for 48 hours. Then post-treatment, these cells were washed. Then it was basically the sterols were extracted by adding chloroform isopropanol 1:2 ratio. Then these samples were centrifuged, and these supernatant contains these sterols were collected in fresh vials. It was basically dried under vacuum. Then in that dried pellet, we have added 200 microliter of chloroform methanol in 1:2 ratio. Then it was basically added with acetonitrile and subjected for rapid fire MS analysis. So this slide is basically, talks about the how this assay is sensitive and how this assay is able to detect the specific sterol. So what we have done was basically we used the commercially available standards of cholesterol, lanosterol, and dihydrolanosterol to understand that how low this assay can detect. The top right-hand side, the top graph, there's a left-hand side, is basically a pure cholesterol standard. So what we have done was basically we used from 3.9 nanomolar to 4,000 nanogram per ml cholesterol were used. If you look at that, the insert, it's basically all the way from this 3.9 to 4,000 nanogram. We see clear linearity in these ranges. And this assay was, what we found was basically this was able to detect about 100 nanogram of cholesterol. So that is basically marked in the arrow. Now, in the adjacent graph, it is basically what we have done was we mixed all these three standard sterols: cholesterol, lanosterol, dihydrolanosterol. So when we applied this method, so what we found was basically this was able to, the cholesterol was able to, the cholesterol MRM was able to pick the cholesterol concentrations from that mixed pool, and it's very similar intensity of the peaks that we were able to get from this mixed as well. Then the same thing we have done with lanosterol. That is basically the bottom graph. That's the bottom left-hand side is basically the pure standard. The adjacent one is basically the mixed one. The same thing is basically with the dihydrolanosterol. So if you look at that, in all these graphs, it is basically all these tested concentrations. It was linear, and for cholesterol, as I said, the minimum detection limit is about close to 100 nanogram. For lanosterol, it was about 250 nanogram. For dihydrolanosterol, it's about 100 nanogram. Now, when we mix or when we keep individual these respective MRM picks respective sterols, when we use the other MRM method, it is basically we were not seeing any response or any, there is no AUC were observed. So that indicates that it is basically this assay, this detection is, this method is very sensitive. It can be able to discriminate at least from these three different sterols. So moving to the next slide, this is basically, this slide talks about the cell density optimization as well as plate format and DMSO tolerance. The left-hand side graph is basically what we have done here was we used three different plate formats. That is basically 6-well plate, 24-well plate, 48-well plate. 6-well plate is basically in red color, and the blue color is basically 24-well plate, and the green one is 48-well plate. Now we used to cell number from 12,500 cells to all the way to 2 million cells. So now what we found here was basically, at 1 million cells. So that's if you look at that, the red arrow, the red bar with the red arrow, it is basically It is a 6-well plate. So using 6-well plate, what we found was it requires 1 million cells to produce the best response. That we got about close to about 7-fold response compared to the background. So when we come to the 24-well plate, it is basically what we found was the right cell density was about 250,000 cells. That is a blue arrow, the blue bar. Now coming to the 48-well plate, so what we found was basically 125,000 cells were able to produce the maximal best response of close to about 8-fold. So from this data, what we identified and what we decided was basically, since the 48-well plate is basically producing the better response or comparable response with the 6-well and 24-well plate, being smaller in size and having a higher throughput, and the requirement of sample is lower, so we decided to use 48-well plate. Now coming to the right hand side, it basically talks about the DMSO tolerance, and this is basically we have tested different concentrations, and we found that 0.5% DMSO is basically tolerable for this assay as well. Now moving to the next slide, it's basically using these optimized parameter. So what we have done was basically we used oxyconazole as a tool compound, that is the left hand side graph, and used all these 3 plate formats, 6-well, 24-well, 48-well plate format, and generated a DRC and calculated the IC50. Now if you look at the bottom table, the IC50s were in the range of around 260 to 290 nanomolar between these 3 plates. That is basically very well in alignment with the reported data from the GCMS. It's about close to 250 plus or minus 40 nanomolar. So what this indicates is basically this assay is, and this is basically we reproduce at least 3 times. So it's all the time. It was very, very consistent, very reproducible, and since actually we were getting better response and better data quality with the 48-well plate, and we used 48-well plate and selected few of the tool compound that is basically assessed from the biochemical assay. The biochemical assay is basically a very different platform. It was used using a purified CYP51A1 enzyme. So based on that data, so we selected highly potent compound, less potent compound, and inactives. If you look at the right hand side, it is basically using the 48-well plate format. This is it's clearly we were able to reproduce this rank order what we observed with the biochemical data. So this assay is basically this clearly indicates that this is, again, it's very robust, very reproducible. To conclude this one, so we have now successfully validated assay for PGD2 as well as assays for these at least these three sterols based on 4 key factors that basically the throughput, quality of the data, the cost effectiveness, as well as the reliability. So this is in all the 4 parameters. All these assays what we have established is basically very well. It is basically it's matching. The ultimate miniaturization of these techniques, as I said in the beginning, currently there are a lot of hindrances are there in using these level-3 technologies, especially the LCMS, because of the less throughput and high cost, high sample requirement. Now, with rapid fire, with this miniaturization, this will help enable the drug discovery researchers to screen new chemical entities with minimally manipulated biological systems with minimal sample volume with a high reproducible data quality. So with this, I end my presentation. I hand over this one to my host for the next Q&A session.
Okay, well, thank you very much. Just a reminder for the audience, if you want to ask questions, you can send them in via the questions widget. Just type them into the box at the top left-hand corner of your screen and click submit. But for now, we have some questions here. So, how long is BGD2 is stable in your assay?
It's a good question. So what we have done was basically, so once it is basically these experiments are done, so we kept the plate for at least 4 to 5 days. Then what we found was basically this PGD2 was stable up to 72 hours in our assay conditions.
Okay, very good. All right, another question here. What is the detection range for PGD2?
Okay, so that in one of the slides, basically I was talking about the lower detection limit for PGD2 in our assay was about 19 nanomolar nanogram per ml, and upper limit of this assay was basically I used up to 4,000 nanogram per ml the standard, and we found that it is basically this assay is able to quantitate, detect up to that level. We haven't gone beyond 4,000 nanogram because we know that the endogenous level of this PGD2 will be very low. So, but I would say that based on our experiment, I would say it's basically about from 19 nanomolar to all the way to 4,000 nanomolar.
Okay, fantastic. All right, another question for you here. What are the endogenous levels of PGD2 and sterols? Were detected in RBL and HC cat cells?
Coming to the PGD2 levels in RBL cells, what we found was upon stimulating with 1 micromolar calcium ionophore, it was about 60 to 80 nanogram per ml. This is what actually we found in the supernatant. Coming to the level of these sterols in Hackett cells, when we inhibit CYP51A1 in the pathway, so what we found was lanosterol was from 600 nanogram per ml to all the way to 900 nanogram per ml. So this is the level that we got for lanosterol in Hackett cells. Coming to the dihydrolanosterol, it's about close to 800 nanogram to all the way to 1300 nanogram per ml. Now the cholesterol was basically when we inhibit the CYP51A1, obviously the cholesterol level goes down. So what we found was basically this was below 100 nanogram per ml. So this is what actually we got from Hackett cells, and the cell number what we used was about 1 million cells. All these numbers actually I'm talking about for 1 million cells. The previous PGD2 level is basically for 50,000 cells, that 60 to 80 nanogram per ml. I hope actually this answers your question.
Okay, well, thank you very much. I'm sure it did. All right, another question asking, Did you try simultaneous detection or quantitation of other prostaglandins?
That's a good question. Actually, this is currently we are now focusing, although actually we built an assay for only for prostaglandin PGD2. But however, as I discussed in my in my presentation, that there are a lot of other prostaglandins very similar in structure, and there are a lot of limitations with the other assay formats. Certainly, we can use this technique and build, and currently that exercise is going on to simultaneously detect other prostaglandin as well from the same cell supernatant. Once the data is available, maybe I can share those.
All right, well, thank you very much. I think that about does it for questions today. So that just leaves me to thank Dr. Prem Kumar for what was a great presentation, and to GVK Biosciences for sponsoring this session. To all attendees, you'll receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. We look forward to sharing further webinars with you, so please do keep an eye out on the website just mentioned. And follow us on Twitter @BRWebinars for daily updates, and join our LinkedIn group, Business Review Webinars. Thank you all once again, and I hope you have a great day.